There is something almost magical about the fact that your blood type was decided before you were even born. It was chosen, in a way, by a conversation between your parents. Not a spoken conversation, but a quiet, biological one, happening in the very first moments of your life. Tonight, we are going to explore why humans have different blood types, what that means, and how this beautiful complexity came to be. And as we travel through this soft science together, I hope you find yourself drifting further and further into rest. Let us begin at the very beginning, not with charts or clinical words, but with the simple, warm image of your blood moving through you right now, slowly, steadily, like a river that has been flowing since before you can remember. Your blood is made of several things: red cells that carry oxygen, white cells that protect you, platelets that help you heal, and a pale golden liquid called plasma that carries everything along. But on the surface of your red blood cells, there are tiny markers. Think of them like small flags or little name tags, molecules that sit on the outside of each cell, gently announcing what type they are. These markers are called antigens, and it is the pattern of these antigens that gives you your blood type. The most well-known system for describing blood types is called the ABO system. In this system, there are 4 possible types: A, B, AB, and O. The letter tells you which antigen, which little marker, is sitting on the surface of your red blood cells. If you are type A, your cells carry the A antigen. If you are type B, your cells carry the B antigen. If you are type AB, your cells carry both. And if you are type O, your cells carry neither. They are O, your cells carry neither. They are smooth and unmarked, like a stone that has been polished by a gentle stream. Scientists discovered the ABO blood group system in 1901. A man named Karl Landsteiner noticed something curious while mixing blood samples in his laboratory. He found that when certain blood types were combined, the cells would clump together, a process called agglutination. It looked like tiny snowflakes forming in the liquid. He realized that blood was not simply blood. It came in types that recognized each other, or didn't. He won the Nobel Prize for this discovery in 1930. But the story of blood types goes back much, much further than any laboratory. The ABO antigens are actually made of sugar chains, tiny molecular structures that sit on the surface of your red blood cells like intricate little sculptures. These sugar chains are built step by step inside your cells, and the instructions for building them are written in your DNA. The gene responsible is called the ABO gene, and it can come in different versions, what scientists call alleles. One version builds the A antigen. Another builds the B antigen. A third version is slightly broken. It cannot build either antigen, which is how you get type O. The gene you inherited for your blood type came from your parents. One copy from your mother, one from your father. This is the same for almost every trait you carry. If you received one A allele and one B allele, you became type AB. If you received one A allele and one O allele, you became type A, because the O allele is recessive, it is quiet, it steps back, and the A allele does the work. If you received 2 O alleles, one from each parent, then you became type O. This beautiful logic is called Mendelian inheritance, named after a gentle monk named Gregor Mendel, who spent his afternoons counting pea plants in a garden. Type O is the most common blood type in the world. About 44% of people have it. This is not an accident. It is the result of thousands of years of human history, of populations moving across continents, of small communities surviving in different conditions. Blood type frequencies are not the same everywhere. In some parts of South America, nearly everyone is type O. In Central Asia, type B is more common. In parts of Europe, type A predominates. These patterns are like fingerprints left behind by ancient migrations, ancient diseases, ancient moments in history that shaped who survived and who thrived. Before we go further into that ancient history, take a breath. Let your shoulders settle. Your body knows how to do this. It breathes without asking you. It circulates without your attention. It keeps everything balanced while you rest. You are safe. You are warm. And there is nothing you need to do right now except listen. There is another important piece of the blood type puzzle, a separate system called the Rh factor or rhesus factor. You might have seen it written as the positive or negative that comes after your ABO type. If you are A positive, you have the A antigen and also a protein called the Rh factor on your red blood cells. If you are A negative, you have the A antigen, but not the Rh protein. The Rh factor was discovered in the 1940s and is named after rhesus monkeys because scientists first noticed this protein in rhesus monkey blood before finding it in humans. About 85% of people worldwide are Rh positive. The remaining 15% are Rh negative. The Rh negative status is more common in people of European descent, and it is thought to have arisen as a genetic mutation at some point in human prehistory. Where and when exactly this happened remains a soft mystery. One of those beautiful, unanswered questions that science holds gently, with curiosity rather than urgency. The Rh factor matters in a particular situation that happens during pregnancy. If a mother is Rh negative and her baby is Rh positive, inheriting the Rh factor from the father, the mother's immune system may, over time, start to recognize the Rh protein as foreign. This is called Rh incompatibility. For many centuries, this was a source of heartbreak that no one understood. Babies would be born too early or too fragile, and no one knew why. It was only in the 20th century that scientists unraveled this mystery and then developed a simple treatment, a gentle injection called Rh immunoglobulin that prevents the problem from developing at all. Medicine, learning to speak the language the body already knew. Now, let us travel even further back, past all the laboratories, past all the textbooks, into deep time, where the story of blood types truly begins. Scientists believe that the ABO blood group system is extraordinarily ancient. When they compare the blood type genes of chimpanzees and gorillas with those of humans, they find the same basic system, the same A, B, and O alleles, existing across millions of years of evolution. This suggests that blood types did not appear in humans. They were inherited from ancestors we shared with the great apes more than 6 million years ago. Think about that for a moment, gently. The blood type flowing through you quietly tonight is not just yours. It is a soft echo of something that existed before the first human ever walked across the open savanna. It is older than language, older than fire, older than any story ever told around any fire. You are carrying something ancient with you, and you carry it as easily as breathing. If you are enjoying this story so far, feel free to leave a comment below and let me know. It means so much, and it helps others find their way here too. And if you would like more stories like this one, do check out the playlist. There are many gentle nights waiting for you there. Why do we have different blood types at all? What is the evolutionary reason for this variety? This is one of the most beautiful questions in all of biology, and the answer involves something called balancing selection, a process where having different versions of a gene in a population is actually more beneficial than everyone having the same version. One major theory involves disease resistance. Different blood types may offer different levels of protection against different illnesses. Over thousands of years, as humans spread across the world and encountered different parasites, viruses, and bacteria, different blood types may have offered advantages in different environments. Type O blood, for example, may offer some protection against the most severe forms of malaria. This might explain why type O is so common in regions where malaria has historically been widespread. Meanwhile, people with type A or type B blood may have had advantages against other diseases, perhaps cholera or certain types of plague. This is not about one type being better than another. It is about the extraordinary wisdom of diversity. When a population has a mix of blood types, it is more resilient. No single disease can sweep through everyone equally. The variety itself is the protection. It is like a forest with many different kinds of trees, more complex, more beautiful, and far harder to destroy. Researchers have studied blood type and disease resistance for decades. Studies have found that people with type O blood may be slightly more susceptible to cholera and to a certain stomach ulcer-causing bacterium called H. pylori. Meanwhile, type A blood may have historically been more vulnerable to smallpox, which was once one of the most devastating diseases on Earth. These are subtle differences, statistical tendencies, not certainties. But over thousands of generations, even small differences in survival can shift the frequencies of blood types across entire populations. Let your eyes grow soft now. Let the room around you settle into its nighttime quiet. There is nothing sharp here, nothing loud, just the slow movement of ideas drifting through you like warm water. The story of blood types is also the story of human migration. Scientists can look at the distribution of blood types around the world and read the history of our species in those numbers. Type B blood, for example, is most common in Central and East Asia and becomes progressively rarer as you move westward toward Europe. This gradient in blood type frequency corresponds beautifully to the ancient spread of human populations from Africa outward, a slow, quiet migration that happened over tens of thousands of years, following rivers and coastlines, moving through grasslands and forests, carrying every possible blood type along. There are places in the world where populations have remained relatively isolated for a long time. Small island communities, mountain valleys, remote plateaus. And in these places, blood type distributions can be very different from the world average. Some indigenous populations of the Americas have almost entirely type O blood. Some groups in the Himalayan highlands have high frequencies of type B. These differences are not just statistics. They are stories written in the ancient language of genetics about where people came from and how they lived. Isn't it extraordinary, in the softest possible way, to think that your blood type connects you to an ancestral journey? That somewhere in the long chain of your family, reaching back through great-grandparents and great-great-grandparents you will never know, someone moved across a continent, survived a winter, outlasted a disease, and passed their blood type forward to you. You are the latest chapter in a story that began in Africa, in the warmth of a very old dawn. The ABO system is the most well-known blood type system, but it is not the only one. Human blood can be classified in many different ways. In fact, scientists have identified over 40 different blood group systems, each one based on different proteins or molecules on the surface of red blood cells. There is the Kell system, the Duffy system, the KID system, the MNS system. Each one a different layer of identity written on your cells. Most of these systems matter primarily in medical settings, especially for blood transfusions, but their existence is a reminder of how breathtakingly complex and individual each human body truly is. The Duffy antigen, for example, part of the Duffy blood group system, is particularly interesting from an evolutionary standpoint. People who lack the Duffy antigen on their red blood cells are almost completely resistant to a type of malaria caused by a parasite called Plasmodium vivax. The absence of the Duffy antigen is extremely common in West African populations, and scientists believe this is a direct result of thousands of years of natural selection in response to malaria. The body, in its quiet wisdom, learned to remove a doorway that the parasite was using to enter. Evolution, working patiently, generation by generation, to keep people safe. And if you would like to share this story with someone who has trouble sleeping, please do. And a like on the video helps so much in letting others discover this little corner of calm. It truly makes a difference. Think for a moment about what a blood transfusion involves and how remarkable it is that we can do it at all. Before Karl Landsteiner's discovery in 1901, doctors had been attempting blood transfusions for hundreds of years with wildly unpredictable results. Sometimes the recipient would recover. Sometimes they would have a severe reaction and die. No one understood why. Some transfusions used animal blood. Doctors in the 1600s tried transfusing lamb's blood into humans, sometimes with surprising short-term success, sometimes with tragedy. It was only once blood types were understood that transfusion medicine became safe and reliable. Today, blood banking is a global system of extraordinary quiet efficiency. Blood is typed and matched before transfusions to ensure compatibility. Type O negative blood, lacking both ABO antigens and the Rh factor, is considered the universal donor blood for red cells because it can be given to anyone in an emergency without risk of a type mismatch reaction. People with type O negative blood are sometimes called universal donors, and blood banks are always especially grateful for their donations. People with type AB positive blood, on the other hand, are called universal recipients for red cells. They can receive blood of any type because their immune system recognizes all the main antigens as self. There is something poetic in that. The body that can accept anything, that sees no stranger in any blood it receives. Your immune system is, in a way, the author of the blood type story. It is the immune system that produced the antibodies, the defensive proteins that react to foreign blood type antigens. If you are type A, your body carries antibodies against the B antigen. If you are type B, you carry antibodies against A. If you are type O, you carry antibodies against both A and B. And if you are type AB, you carry neither, which is why you can receive anyone's blood. These antibodies form early in life, not because you have ever been exposed to foreign blood, but because bacteria and other microorganisms in your environment carry molecules that resemble the ABO antigens. Your immune system encounters these environmental triggers in infancy and learns to produce the appropriate antibodies. It is one of the many ways your body is constantly learning, constantly building its own library of recognition. Let the weight of your body settle more deeply now. Feel the softness beneath you. Somewhere in your chest, your heart is beating in its steady, tireless rhythm, sending blood to every corner of you, nourishing every cell. It does this all night, without being asked. It does this while you sleep. There is a long history of human fascination with blood as something meaningful, something that carries identity and connection. Many ancient cultures believed that blood carried personality, courage, spirit. The Japanese tradition of blood type personality theory, which associates type A with being organized and careful, type B with being creative and individualistic, type O with being confident and social, and type AB with being complex and thoughtful, remains popular in Japan today, even though scientists have not found strong evidence to support these personality associations. The appeal makes sense, though. We want our bodies to tell us something about who we are. And in a way, they do. Just not necessarily in the way popular culture imagines. What blood types do genuinely tell us is something about history, about ancestry, about the invisible connections between all living humans. When researchers study ancient DNA, carefully extracted from the bones of people who lived thousands of years ago, they can sometimes determine the blood type of those ancient individuals. A Neolithic farmer in Europe, a Bronze Age warrior in the steppes, a person who lived beside the Nile 5,000 years ago. And when they find a blood type that matches yours, there is a soft thrill in it, a quiet recognition across an enormous span of time. The science of blood types has also touched the study of ancient populations in profound ways. Researchers studying the remains of passengers from the Titanic, or soldiers from ancient battlefields, or people buried in medieval plague pits, have used blood type analysis as one small piece of the puzzle of who those people were. Blood type has become a kind of quiet witness to history, patient, biological, never lying. In more recent research, scientists have begun exploring whether blood type might influence more than immune response, whether it might play subtle roles in cardiovascular health, cognitive function, or the risk of certain conditions. Some studies suggest that people with type A/B blood may have a slightly higher risk of memory difficulties later in life, possibly related to a clotting factor associated with the ABO gene. People with type O blood may have slightly different risk profiles for certain conditions compared to those with type A or B. These are early findings, tendencies visible only across large populations, not destinies written in stone. Your blood type is one small thread in the vast, beautiful weaving of your health and your life. If you are curious about your own blood type and do not know it, you can often find out through a simple test. Many blood donation centers will tell you your type when you donate. It is a small thing to know about yourself, but it carries that lovely weight of ancient story. We are settling deeper now, aren't we? The science is becoming softer, more like music than information. Let it wash over you. You do not need to remember any of this. It is simply here to keep your mind gently occupied while your body does what it knows how to do. Drift toward sleep. There is one more layer to the blood type story that deserves a quiet moment. The genetics of it all, and how beautifully simple and profound it is. Your ABO blood type is determined by a single gene on chromosome 9. One gene. Out of the approximately 20,000 genes in your entire human genome, just one is responsible for whether you are type A, B, AB, or O. And yet, through the mechanism of natural selection, through the long pressure of disease and migration and survival, this one gene has been shaped and sorted across billions of people over millions of years, leaving behind the extraordinary global tapestry of blood type distribution that exists today. Chromosome 9 is one of the medium-sized chromosomes, not the largest, not the smallest. It carries thousands of other genes as well, involved in everything from metabolism to the development of your nervous system. The ABO gene sits quietly among its neighbors, doing its one small job, instructing your red blood cells on what kind of antigen to wear. And it does this job every single moment of your life, in every one of the trillions of red blood cells your body produces. Your bone marrow, the soft, spongy tissue inside your larger bones, produces red blood cells continuously at an almost incomprehensible rate. About 2 million new red blood cells are born in your bone marrow every single second. Each one emerges already wearing the antigen that your ABO gene specified, already carrying your blood type, already part of the ancient story. They will live for about 120 days, circulating through you, carrying oxygen, and then be quietly recycled. New ones will take their place. The river keeps flowing. It is worth pausing to appreciate how quietly extraordinary your body is, even in this ordinary moment of lying still and preparing for sleep. Without any direction from you, your bone marrow is producing new blood cells. Your heart is beating. Your immune system is patrolling. Your blood type antigens are sitting on the surface of your cells, patient as stones in a river, doing what they have always done, what they were doing before your grandparents were born, what they were doing before the first human cities, before the first human language, ancient, quiet, faithful. Do feel free to leave a comment if this is your first time here. It is always wonderful to hear from new listeners, and the playlist has many more nights like this one, waiting whenever you need them. The story of blood types is also, in a lovely way, a story about what makes you specifically you. Because while your ABO type places you in one of 4 broad categories, the full picture of your red cell surface is far more individual than that. When you combine all 40+ blood group systems together, all the different antigens, all the different possible combinations, the probability of any 2 people having exactly identical red blood cell surfaces is extraordinarily low. Each blood type profile is almost like a fingerprint. You, in your biological specificity, are genuinely one of a kind. There is a beautiful concept in immunology called self and non-self. Your immune system spends the first months and years of your life learning to recognize everything that is you, every molecule, every protein, every antigen on every one of your cells. It memorizes the map of your body, and then, with quiet vigilance, it watches for anything that does not belong to that map. This is why blood type matching matters in transfusions. Because if a foreign blood type enters you, your immune system may recognize the unfamiliar antigens and respond. But when matched blood flows into you, it is recognized as self, accepted, welcomed, absorbed into the river without a ripple. In a way, your blood type is one of the first things about you that your immune system ever learned. From the very beginning, before you had memories or language or dreams, your immune system was reading your blood type and saying, yes, this is us. This belongs here. There is something quietly moving about that. Let your breathing slow a little more now. Let the night gather softly around you. There is nothing you need to solve or remember or hold on to. The story is here, flowing around you like warm air. One of the most fascinating recent developments in blood type research involves a discovery made in 2022 when scientists at the University of Bristol identified the molecular basis of a very rare blood type called OR, solving a mystery that had puzzled researchers for over 50 years. A woman in the 1980s had experienced complications during pregnancy, and doctors noticed she had unusual antibodies against a blood antigen they did not recognize. It took decades of careful science, culminating in advanced genomic techniques, to finally identify the gene responsible for the ER blood group system. It was a beautiful example of science's long patience. Some questions wait decades to be answered, sitting quietly in the literature like seeds waiting for rain. There are also extremely rare blood types, some so rare that only a handful of people in the entire world have them. The rarest of all is sometimes called the golden blood type, or Rh null. People with Rh null blood have none of the Rh antigens on their red blood cells. None at all, out of the more than 50 known Rh antigens. As of recent counts, fewer than 50 people in the world are known to have this blood type. They are sometimes called golden blood donors because their blood is extraordinarily valuable for transfusions in rare cases. Imagine carrying something that rare inside you, something that fewer than 50 people on Earth share. And yet, those people live perfectly ordinary lives, unaware most of the time that their blood is writing a particularly unusual sentence in the story of human biology. At the other end of the spectrum, type O negative, the universal donor, is carried by roughly 7% of the global population. These people, too, carry something quietly special inside them: the ability to help anyone who needs blood in an emergency. It is one of the more beautiful consequences of blood type biology, that a genetic variation that arose tens of thousands of years ago now makes certain people particularly precious in moments of crisis. The night is deepening around you now. Your muscles are soft. Your thoughts are becoming less sharp, more like drifting clouds than clear images. This is exactly where you want to be. Research into blood types and health continues to unfold in gentle ways. Scientists studying COVID-19 found some evidence that people with type O blood may have had slightly different susceptibility to certain severe outcomes compared to those with other blood types, though the mechanisms are still being studied and the differences are modest. This echoes earlier findings about blood type and other infectious diseases, adding another soft layer to the story of why blood type diversity exists. There is also research into blood type and gut microbiome, the community of bacteria that lives in your digestive system. Some studies suggest that blood type antigens, or molecules related to them, may influence which bacteria thrive in your gut. Since the ABO gene is expressed not only on red blood cells, but also on the cells lining your digestive tract, the antigens on those cells may act as a kind of selective surface, welcoming certain bacterial species and not others. Your blood type, in other words, may be quietly shaping the invisible ecosystem living within you. Science also tells us something tender about blood types and family. Because blood type is inherited according to simple genetic rules, it has long been used before the era of DNA testing to provide soft evidence about biological relationships. A child can only have a blood type that is genetically possible given their parents' types. This simple fact has been woven into countless personal stories, moments of recognition, of connection, of gentle understanding between parents and children. Blood carrying family forward, generation after generation. And in the most literal sense, that is exactly what blood does. It carries the oxygen and nutrients that every cell in your body needs to survive. Without that quiet circulation, nothing else would be possible. Not thought, not movement, not love, not sleep. Blood is the medium through which your body sustains itself moment to moment, breath to breath, heartbeat to heartbeat. If this story has brought you a little closer to sleep, a comment below would make my night. And if you know someone who might enjoy drifting off to science, please share this with them. It means everything. Your heart beats about 60 to 100 times per minute at rest. In a single day, it beats more than 100,000 times. Over a lifetime of 70 years, your heart will beat approximately 2.5 billion times. And in every one of those beats, it is pushing your blood with your blood type, your antigens, your ancient inheritance, through the 60,000 miles of blood vessels that thread through your body. 60,000 miles. That is more than twice the circumference of the earth. All of it inside you. All of it working quietly while you sleep. The blood vessels themselves are extraordinary. From the large aorta, wide as a garden hose, to the capillaries so narrow that red blood cells must pass through them single file, gently bending their flexible bodies to squeeze through. In those capillaries, just a few cells across, the work of breathing happens at the cellular level. Oxygen released, carbon dioxide picked up, the exchange so small and so constant that it is almost unimaginable. And yet, it is happening in you right now, everywhere in your body as you rest. Think of the capillaries in your fingertips, blood flowing all the way out to the very edges of you, carrying warmth, carrying oxygen, carrying your blood type markers on every red cell. The cells that visit your fingertip have the same antigen as the cells near your heart, the same genetic instruction, the same ancient origin. Every cell consistent with every other, the same quiet identity expressed everywhere at once. We are near the end of our story now. Though the night is still full and soft around you, let the science settle like snow. not demanding to be remembered, just gently present. You have learned tonight about sugar chains on the surfaces of cells, about ancient genes shared with great apes, about malaria and migration, and the quiet genius of genetic diversity. You have traveled across millions of years and across every continent, all from wherever you are lying now. And through all of it, your blood has been doing what it always does, flowing, carrying, sustaining, connecting. It was doing this before you started listening tonight. It will keep doing it long after you fall asleep, the river that is you, flowing quietly in the dark. Blood type research is still very much alive. Scientists are exploring new blood group systems, discovering the functions of rare antigens, developing better methods for matching blood for transfusions in people with complex medical needs. There are ongoing studies into the precise evolutionary pressures that shape the global distribution of blood types. There are questions still waiting to be answered, gently, patiently, by researchers in laboratories around the world who find in the humble red blood cell a universe of discovery. And somewhere out there tonight, in a blood bank in a city you have never visited, bags of carefully typed blood are waiting in quiet refrigerators. A positive, B negative, O positive, AB negative, each one labeled, each one ready to help someone. Thousands of people donating something they will easily replace, offering a temporary gift of their ancient inheritance to someone who needs it. It is one of the most ordinary, extraordinary things humans do for each other. Your blood type is not the most important thing about you, but it is one of the oldest things about you. It connects you to your parents, to your grandparents, to ancestors whose names you do not know, to a moment deep in evolutionary history, when the first primate version of the ABO gene arose and was passed forward. It connects you to the 6 million people on Earth who share your specific blood type combination, scattered across continents, living their lives, unaware of this quiet bond. And it connects you tonight to the simple fact that you are alive, that a heart is beating in your chest, that blood is moving through you, that your body is real and present and warm in this moment. There is something worth sitting with for a moment. The idea that your blood type, this quiet detail written into every red cell in your body, is not really yours alone. It never was. It came to you through a chain of people stretching back so far that the earliest links in that chain were not even fully human yet. It passed through your parents, through their parents, through generations of people who lived through winters you cannot imagine, who crossed landscapes that no longer exist, who survived illnesses that have no name anymore. And now it rests in you softly, without any fuss, doing its small, faithful work. Scientists who study evolutionary genetics use a term called coalescence, the idea that if you trace any gene backward through time, all the different versions of it will eventually meet at a common ancestor. For the ABO gene, that coalescence point, the moment when all the different blood type alleles trace back to a single ancestor population, is estimated to be millions of years in the past, long before modern humans existed. This means that when you meet someone with a different blood type, you are not so different after all. You simply inherited different versions of the same ancient gene, shaped by different pressures, different places, different moments in the long story of life on Earth. The B allele, which gives rise to blood type B, appears to have spread westward from Asia into Europe much later than the A allele, which is older and more widespread. Some researchers believe the B allele may have increased in frequency during the great pandemics of the medieval period, since it may have offered some degree of protection against certain infections. The plague that swept through Europe and Asia in the 14th century, carried by bacteria spread through rats and fleas, may have quietly shifted the blood type distribution of entire regions by altering which people survived and had children. Terrible moments in history, leaving their mark in the softest biological language. Bubonic plague, caused by a bacterium called Yersinia pestis, is one of the most studied diseases in the context of blood type research. There is evidence that people with type O blood may have been somewhat more vulnerable to plague than people with other types, which could explain why certain European populations, historically ravaged by multiple waves of plague, show lower frequencies of type O compared to populations that were less affected. History written in blood type frequencies, patient as sediment. It is a humbling thought, and also a quietly comforting one. The fact that you exist, that you are here tonight, breathing, resting, listening, means that every single one of your ancestors, all the way back to the very beginning, survived long enough to have children. Every one of them made it through whatever difficulty their time and place presented. You are, in a literal biological sense, the product of an unbroken chain of survival, and your blood type is one of the small molecular badges the chain has carried forward to you. Let the weight of that settle gently. You come from survivors, and tonight you are doing the simplest, most restorative thing a survivor can do. You are resting. Blood type research in recent decades has been transformed by the tools of genomics, the ability to read DNA sequences quickly and cheaply, to compare genomes across thousands of individuals, to find patterns in vast biological data sets that no single researcher could ever have spotted alone. These tools have allowed scientists to revisit old questions about blood types with new precision, and to ask new questions that were not even imaginable before. One area of active research involves something called genome-wide association studies, or GWAS. In these studies, scientists look across the entire genome of thousands or even hundreds of thousands of people, searching for regions of DNA that are statistically associated with particular traits or health outcomes. The ABO gene region has shown up in many of these studies, associated with a remarkably wide range of things, from cardiovascular risk factors to certain cancer susceptibilities, to levels of various proteins in the blood. Some of these associations are well understood, others remain intriguing puzzles. The ABO gene, it seems, does not simply sit quietly doing its one job. It has relationships with other parts of biology that are still being mapped. For example, the ABO gene influences the levels of a molecule called von Willebrand factor in your blood, a protein that plays an important role in clotting. People with type O blood tend to have lower levels of von Willebrand factor than people with type A, B, or AB. This means people with type O blood may bleed a little more easily in some circumstances, but may also have a slightly lower risk of certain types of blood clots. Neither of these is a dramatic difference in everyday life, But across populations and across decades, these small tendencies add up to patterns that researchers can detect and study. Blood clotting is itself a magnificent biological story. When you are injured, even just a small cut on your finger, your body launches an intricate cascade of reactions involving more than a dozen different proteins, all activating each other in a precise sequence, ultimately producing a clot that seals the wound. This cascade is sometimes called the coagulation cascade, and it has been refined by hundreds of millions of years of evolution. Von Willebrand factor is one of the early players in this cascade, and the fact that your blood type influences its level is a reminder of how deeply interconnected all the systems of your body are. There are no isolated parts. Everything speaks to everything else. Feel that interconnection in yourself right now, if you like. The way your breathing influences your heart rate. The way your heart rate influences your sense of calm. The way your sense of calm allows your muscles to release. The way your muscles releasing lets your body sink more deeply into rest. Everything speaking to everything else. A whole system tending toward quiet. There is a researcher whose work has added a great deal to our understanding of blood type evolution, a scientist named Laure Segurel, who has spent years studying the evolutionary history of the ABO gene across primates and human populations. Her work has helped clarify just how ancient the different blood type alleles are, and has suggested that the maintenance of blood type diversity in human populations is likely the result of multiple different selective pressures acting over very long periods of time. Not one single advantage, but many overlapping ones, shifting in importance as environments and diseases changed. The picture that emerges is not a simple one, but it is a beautiful one. A gene shaped by the full complexity of human history, carrying within it the echoes of every landscape and every epidemic our species has ever encountered. Another researcher, Peter D'Adamo, became well known in the 1990s for proposing the idea that people should eat according to their blood type, the so-called blood type diet. While this idea captured a great deal of popular imagination, the scientific consensus has not supported it. Large studies comparing dietary outcomes across blood types have not found evidence that eating patterns matched to blood type produce better health outcomes. Blood type influences some aspects of physiology, but it does not appear to determine which foods nourish you. The story of blood types is fascinating enough without needing to extend it into areas where the evidence does not point. Science is comfortable with limits. It finds them clarifying, not disappointing. If you are still awake and following along, a gentle thank you for being here. And if you feel moved to leave a comment about what you find most fascinating about tonight's story, that would be a lovely thing to wake up to. And please do explore the playlist if you would like more nights like this one. Let us travel now into the world of blood type and the very beginning of human life, the moment of conception and the quiet drama that can unfold when blood types interact across the boundary between a mother and her baby. We touched on Rh incompatibility earlier, but the story is richer than a single detail. When a baby grows in the womb, small numbers of fetal cells can sometimes cross the placenta and enter the mother's bloodstream. This is a normal part of pregnancy, a kind of quiet biological conversation between two bodies sharing space. For most blood type differences, this causes no difficulty. But for certain combinations, particularly when the mother is Rh negative and the baby is Rh positive, the mother's immune system may, over time, produce antibodies against the Rh factor. These antibodies can then cross back through the placenta in a subsequent pregnancy and reach the fetal bloodstream, where they may cause problems for a baby's red cells. This condition is called hemolytic disease of the fetus and newborn, or HDFN. For most of human history, it was a mystery. Babies born pale and weak, sometimes not surviving the first days of life, and no one understanding why. In the 20th century, as blood type science advanced, the mystery unraveled. And then, beautifully, a solution was found, a simple injection given to Rh negative mothers during and after pregnancy, which prevents the immune reaction from developing in the first place. The injection contains Rh immunoglobulin, antibodies that gently neutralize any fetal Rh positive cells before the mother's own immune system can respond to them. A preventive whisper stopping the problem before it begins. This treatment, sometimes called the Rogum injection, has saved countless lives since its introduction in the 1960s. It is one of the great quiet successes of 20th century medicine, a solution born directly from understanding the biology of blood types, knowledge turning into care, science becoming gentleness. HDFN can also occur with ABO incompatibility, though it is usually milder. If a mother is type O and her baby is type A or B, the mother's naturally occurring antibodies against A and B antigens can occasionally cause mild hemolysis, the breakdown of the baby's red cells, after birth. This is typically manageable and usually resolves on its own, the body navigating its own complexity with resilience. There is something deeply moving about the biology of pregnancy and blood type when you look at it from a certain angle. Two blood types, two sets of antigens, two immune systems, mother and baby, negotiating across the thin membrane of the placenta for 9 months. The mother's immune system must perform an extraordinary feat. It must remain tolerant of the baby, which carries foreign proteins from the father, while still defending the mother against genuine threats. This tolerance is achieved through a complex set of immunological mechanisms that scientists are still working to fully understand. The womb is a place of profound biological diplomacy. The night is very soft now. Your breathing is slow. Let the science become less like thinking and more like dreaming. Ideas moving through you without asking to be held. Blood type is expressed not only on red blood cells, but on many other cell types throughout your body. The ABO antigens can be found on white blood cells, platelets, and on the cells lining the blood vessels in many organs, the heart, the lungs, the kidneys. In secretors, about 80% of the population, ABO antigens are also released into body fluids, including saliva, tears, and breast milk. These people are called secretors because they secrete their blood type antigens into their fluids. The remaining 20% are non-secretors, who have a slightly different version of a related gene that keeps the antigens confined to cell surfaces. This secretor status is controlled by a separate gene called the FUT2 gene, and it has its own evolutionary story. Being a non-secretor appears to offer protection against certain gut infections, particularly norovirus, the common stomach bug that spreads so easily in enclosed spaces. The norovirus attaches to ABO antigens in the gut lining to gain entry to cells, and non-secretors who have fewer of these antigens on their gut cells may be more resistant. Nature, as always, finding its own quiet balances. The idea that blood-type antigens in saliva could once have been used forensically is a fascinating one. For much of the 20th century, forensic scientists could determine the blood type of a secretor from a small sample of saliva, sweat, or other body fluids, useful in criminal investigations long before DNA analysis became available. Blood type was one of the first biological markers used to narrow down the identity of an unknown person or to exclude a suspected individual from a crime scene. Science in service of justice, using the quiet language of antigens. In transfusion medicine, the science of blood type matching has grown increasingly sophisticated. For patients who require many transfusions over their lifetime, such as those with sickle cell disease or those undergoing treatment for certain cancers, it is not enough to simply match ABO and RH type. Extended matching across multiple blood group systems helps prevent the development of new antibodies that could complicate future transfusions. This is a careful, patient science, building up a detailed portrait of a person's red cell surface, matching it as precisely as possible with donor blood, minimizing the immune response, maximizing the benefit of each transfusion. Sickle cell disease deserves a moment here because it is connected to blood type history in a poignant way. Sickle cell disease is caused by a mutation in the hemoglobin gene, the gene that makes the protein carrying oxygen inside red blood cells. The mutation causes red cells to become stiff and sickle-shaped under certain conditions, blocking small blood vessels and causing pain. But carriers of just one copy of the mutation, those who do not have the full disease, have a significant degree of protection against severe malaria. This is why the sickle cell mutation became so common in malaria endemic regions of Africa, the Mediterranean, and the Middle East. Like the ABO blood type story, it is a story about the evolutionary bargain struck between genetic variation and infectious disease, a bargain that sometimes brings suffering and sometimes brings survival, and is always far more complex than it first appears. Please do share tonight's story with anyone you know who might be lying awake right now, thinking too hard, needing a gentle voice. And if you are enjoying this journey through the science of your blood, a like on the video is a warm and welcome thing. Let us spend some time now with the remarkable history of blood transfusion, because it is one of the great adventure stories of medicine, full of wrong turns and sudden illuminations, and it ends in the quiet miracle of modern blood banking. The first documented attempt at a blood transfusion in a human being took place in 1667, when a French physician named Jean-Baptiste Denis transfused a small amount of lamb's blood into a young man suffering from a fever. The patient survived, probably because only a small amount of animal blood was used, not enough to trigger a fatal immune response. Denis tried the procedure on several more patients. One died, and Denis was accused of murder, though he was eventually acquitted. France, and then most of Europe, banned blood transfusion for more than a century. The practice was revived in the early 19th century, when a British obstetrician named James Blundell became concerned about women dying from blood loss after childbirth. He developed a device for transfusing blood between humans, avoiding the animal blood problem, and performed the first successful human-to-human transfusion in 1818. Of the 10 transfusions Blundell performed, about half succeeded. He did not know why some worked and some did not. The missing piece, of course, was blood type. Once, Karl Landsteiner described the ABO system in 1901. The practice of blood transfusion was transformed. Within a decade, surgeons were typing blood before transfusions. In World War I, blood transfusion became a genuine life-saving tool on the battlefield, though the logistics of keeping blood available were still challenging. It was not until 1914 that scientists discovered that adding sodium citrate to blood prevented it from clotting, allowing it to be stored. This single discovery, the anticoagulation of stored blood, made blood banking possible. The first blood banks were established in the 1930s in Chicago and in Leningrad, nearly simultaneously. During World War II, blood banking scaled up enormously. Millions of units of blood were collected, processed, typed, and distributed to military hospitals around the world. It was during this period that the concept of voluntary blood donation, giving blood as a civic act, a gift to strangers, became embedded in the culture of many countries. The Red Cross played an enormous role in this, coordinating donation drives and establishing the infrastructure that modern blood banking still rests on. Today, the global blood supply involves approximately 118 million blood donations per year, according to the World Health Organization. Each donation is tested, typed, separated into components, red cells, plasma, platelets, and distributed to hospitals and clinics where it is matched to patients in need. It is one of the largest and most complex logistics operations in all of medicine, running quietly and continuously every day of the year, saving millions of lives that most people never hear about. Your own blood type sits somewhere in that global system, a category, a compatibility profile, a set of molecular markers that would allow a stranger's generosity to reach you in your moment of need. It is a network of care built on the quiet science of antigens, running like a river beneath the surface of daily life. The room is very still now. Let your thoughts drift like leaves. On slow water, there is nothing you need to hold. Everything is already taken care of. One of the most elegant things about blood type genetics is how it illustrates the concept of codominance, a pattern of inheritance where two alleles are both fully expressed, neither one dominating the other. In most familiar genetic examples, one allele is dominant and masks the other. But in blood type AB, something different happens. Both the A allele and the B allele are fully expressed. The red blood cells of a type AB person carry both the A antigen and the B antigen on their surface. Not a blend of the two, but both simultaneously, side by side. It is a small but beautiful example of biological democracy. This codominance is why type AB is considered the universal recipient for red cells in emergency transfusions. The AB immune system already recognizes both A and B antigens as self, so it does not mount an attack against either. And it is also why type AB plasma is the universal donor plasma. Plasma from a type AB person lacks the anti-A and anti-B antibodies that would react against other blood types, making it safe for anyone to receive in plasma form. The same blood type, universal in two different directions at once. Plasma transfusions matter enormously in medicine. Plasma, the liquid component of blood, pale, golden, and rich with proteins, carries clotting factors, antibodies, and many other essential molecules. Plasma transfusions are used in patients with clotting disorders, severe burns, liver disease, and many other conditions. Fresh frozen plasma, separated from donated blood and frozen quickly to preserve its proteins, is one of the most commonly used blood products in hospitals worldwide. Every time you hear that type AB donors are needed, this is part of why. Platelets, the tiny cell fragments that help form clots, also need to be typed and matched, though the rules are slightly different from red cell transfusions. Platelet transfusions last only a few days after donation, which is why blood banks are constantly seeking new donors. The short shelf life of platelets means the supply must be continuously renewed, a living river, always moving. Blood science has also given us insights into organ transplantation. When a new organ is transplanted into a patient, blood type compatibility between donor and recipient is one of the important factors in determining whether the transplant will be accepted or rejected. The ABO antigens are present on the cells of many organs, not just blood cells, so a blood type mismatch between donor and recipient can trigger rejection. This is why transplant matching takes blood type, among many other factors, into careful consideration. The same quiet molecular markers that Karl Landsteiner noticed clumping in a test tube in 1901 are still shaping the decisions of transplant surgeons today. Do feel free to pause and leave a comment if you are finding yourself more awake than you expected. Sometimes science has that effect, the way it opens small windows of wonder right when you thought you were ready to close your eyes. That is all right. The rest will come. And the playlist is always there, waiting whenever you need another night like this one. The future of blood type science holds some extraordinary possibilities. One of the most exciting is the development of what researchers call universal red blood cells. Red cells that have been enzymatically stripped of their ABO antigens, making them compatible with any recipient. Scientists have identified enzymes discovered in bacteria found in the human gut that can efficiently remove both A and B antigens from red blood cells in vitro, converting them to a type O-like state. If this process can be made reliable and cost-effective at large scale, it could dramatically simplify blood banking, reducing the need to maintain separate inventories of different blood types and making emergency transfusions safer and faster. Research published in Nature Microbiology in 2018 described a particularly promising pair of enzymes from gut bacteria that could convert type A blood to type O with high efficiency. The work was led by researchers at the University of British Columbia and caused considerable excitement in the transfusion medicine community. Moving from laboratory demonstration to clinical reality takes time, but the direction is clear and the science is advancing. There is also ongoing work on synthetic blood and blood substitutes, molecules designed to carry oxygen through the bloodstream in place of, or in addition to, natural red blood cells. These hemoglobin-based oxygen carriers, or HBOCs, have been studied for decades with mixed results. The challenge is to create something that performs the oxygen-carrying function of red blood cells without causing unwanted side effects and the complexity of natural blood is a formidable standard to meet. But the research continues, patient and methodical, driven by the hope of one day having a universally compatible, shelf-stable blood product that could be used anywhere in the world without the need for blood typing. The body's own complexity keeps setting the bar higher. Every time researchers think they have found a sufficient substitute for some component of blood, they discover new functions and subtleties that the substitute does not capture. Blood is not simply a delivery system for oxygen. It is a communications network, an immune patrol, a temperature regulator, a pH buffer, a carrier of hormones and nutrients and signaling molecules. To replicate all of that, even partially, is an enormous challenge, one that scientists approach with the same quiet patience that blood itself embodies. Steadily, without hurry, one small discovery at a time. The night has been generous with its science tonight. Let it now be generous with its silence. You have traveled a long way through the story of your blood. From the ancient gene shared with great apes, through the slow spread of human populations across continents, through the quiet evolutionary bargains between blood type and disease, through the history of transfusion medicine, through the soft biology of mothers and babies and immune systems learning diplomacy, all the way to the laboratories where scientists are learning to strip antigens from cells and reimagine what blood can be. And through all of it, the essential fact has remained unchanged. Right now, in this moment, your blood is flowing through you. It is carrying oxygen to your brain, which is softening its activity with every breath. It is carrying warmth to your hands and your feet. It is doing what it has always done, what it was doing before you knew its name or understood its story. Let your body be exactly what it is tonight, a living thing, ancient and complex and quietly miraculous, needing nothing more right now than to rest. There is a particular quality to deep sleep that scientists find beautiful and strange in equal measure. During the deepest stages of sleep, your brain produces large, slow electrical waves called delta waves. The slowest, most synchronized activity your brain ever generates while you are alive. In these moments, blood flow to the brain actually decreases slightly, and the cerebrospinal fluid that cushions your brain and spinal cord pulses in and out in rhythmic waves, washing away metabolic waste that is built up during the day. Sleep is, in a very real sense, a cleansing. The brain doing its housekeeping while the rest of you is still, and your blood carrying your ancient blood type markers on every red cell plays its part in sleep as well, delivering the oxygen and nutrients that support this restoration, removing carbon dioxide, maintaining the delicate biochemical balance that allows deep sleep to happen at all. Even in rest, you are being actively tended by your own biology. Nothing in you is ever truly still. The glymphatic system, the brain's waste clearance network, was only properly described by scientists in 2013 in work led by researcher Maiken Nedergaard at the University of Rochester. Before that discovery, scientists did not fully understand how the brain cleaned itself. Now, we know that during sleep, the spaces between brain cells expand, allowing cerebrospinal fluid to flow more freely through the brain tissue, flushing out proteins and other waste products that have accumulated. This nighttime cleaning is now thought to be important for long-term brain health. sleep is not passive. It is one of the most active and important things your body does. Blood type does not directly influence the glymphatic system as far as current science knows. But everything in the body is connected, and the nutrients and oxygen that the blood carries are what make all of it possible. The blood and the brain in quiet partnership, night after night, doing the work that keeps you well. When you wake tomorrow, you will carry the same blood type you carried tonight, the same antigens on the same red blood cells, the same ancient inheritance, the same quiet connection to your ancestors and to the millions of people around the world who share your type. Nothing about you will have changed in that fundamental way, but your brain will have cleaned itself. Your cells will have rested and repaired. Your immune system will have processed and consolidated, and you will be, in ways both subtle and profound, more ready for the day than you were when you closed your eyes. Sleep is one of the gifts that evolution gave us. Across almost every species that has a nervous system, some form of rest and recovery is necessary. Even fruit flies sleep. Even zebrafish sleep. The need for rest appears to be one of the most ancient features of animal biology, as old in its own way as the need for blood. Rest and circulation, sleep and flow, the two rhythms that sustain you. There is a warmth in knowing that, in knowing that your body was designed for this, that lying still in the dark, letting your mind go soft, allowing yourself to drift, is not laziness or indulgence, but a biological necessity as profound as eating or breathing. You are not doing nothing right now. You are doing one of the most important things a living body can do. If you have made it this far into the night, something in you is very close to sleep now. Perhaps your eyes have been closed for a while. Perhaps the words are no longer forming distinct images, just shapes of sound and warmth. That is exactly right. You are exactly where you should be. The science of blood types will continue to unfold in the years and decades to come. Researchers will find new associations, new evolutionary stories, new clinical applications. The 40-plus blood group systems will be more fully characterized. The universal red blood cell may become a clinical reality. The connection between blood type and the microbiome will be mapped in greater detail. New functions of the ABO gene, beyond antigen production, will be discovered. But none of that needs to happen tonight. Tonight is for resting. Tonight is for the slow work of sleep, which is the body's own science, just as ancient, just as elegant, just as worthy of wonder as anything that happens in a laboratory. Your blood knows how to flow without being asked. Your heart knows how to beat. Your brain knows how to dream. You know how to rest. Let the last of the day's thoughts dissolve now. Let the words go. Let even the soft curiosity that has carried you through tonight's story fade into the background, quiet as a radio playing in another room, barely audible, then gone. There is only warmth now, only the gentle dark, only the slow, even rhythm of your breathing and the deeper, quieter rhythm of your heart. You are safe. You are held. The night is around you like something soft and familiar. Your ancient blood is flowing. Your body is doing what it was made to do. Sleep now. Sleep well. And thank you, truly, for spending this night with the Sleepy Science Channel. The playlist is always here whenever the night needs a little quiet company. Until next time, There is a field of science that sits at the quiet intersection of genetics and history, a discipline called archaeogenetics, or ancient DNA research. In the last 2 decades, it has transformed what we know about human prehistory, and blood types have played a small but luminous role in that transformation. When researchers extract DNA from ancient bones and teeth, from people who lived thousands of years ago, whose names are lost, whose faces we can only imagine, they can sometimes determine the blood type of those individuals with reasonable confidence. And those ancient blood types, compared to the distribution of blood types in modern populations, can tell us something about the movements and replacements of human groups across time. One of the most striking findings of ancient DNA research is how dramatically the genetic composition of Europe changed across different prehistoric periods. The hunter-gatherers who lived in Europe before the arrival of agriculture, people who hunted reindeer and gathered berries and painted on cave walls, had a very different genetic profile from the farmers who arrived from Anatolia around 9,000 years ago, and different again from the steppe pastoralists who swept into Europe from the east around 5,000 years ago. Each of these migrations brought different gene variants, different disease histories, different blood type frequencies. The Europe of today is a layering of all three, and more besides, a palimpsest of migrations and meetings, written in DNA and expressed, among other things, in blood type distributions. Researchers working on the ancient DNA of Neolithic European farmers have found a relatively high frequency of blood type A, consistent with the idea that type A may have spread into Europe with the agricultural transition. Hunter-gatherers, by contrast, appear to have been more frequently type O. The steppe pastoralists brought their own mix. Layer by layer, wave by wave, the modern European blood type distribution was assembled, not by design, but by the accumulation of thousands of years of human movement, survival, and reproduction. It is extraordinary to imagine. The type A blood flowing through someone in central France today may carry, in its quiet molecular language, the echo of a Neolithic farmer who crossed from Anatolia 9,000 years ago, crossing the same sea that ferries tourists today, not on a boat with an engine, but on a simple raft, carrying seeds and goats and a set of blood-type antigens that would still be present 9,000 years later in their descendants' veins. You are part of that long procession. Whatever your blood type, it came to you through a chain of people who moved and survived and loved and had children across an almost incomprehensible span of time. Tonight, you are the furthest point in that chain, the most recent chapter. And you are resting, which is exactly what every link in that chain needed to do every night of their lives to keep going. Let that be a comfort. Rest is not new. Rest is ancient. Every ancestor you have ever had knew this same need, this same surrender to the dark, this same slow unraveling of the day. You are in good company. Ancient blood type detection has its limits, of course. DNA degrades over time, and the older the sample, the more fragmentary the genetic information tends to be. But improvements in laboratory technique have pushed back the horizon of what is possible. Researchers have successfully extracted and analyzed DNA from bones more than 400,000 years old. And while complete blood type determination from such ancient samples remains challenging, the field is advancing steadily. Each year, the window into our deep past opens a little wider, and the story of blood type in human prehistory becomes a little more detailed. One remarkable study published in the early 2020s analyzed the blood types of ancient Egyptian mummies, individuals who lived along the Nile thousands of years ago. The findings showed blood type distributions that were broadly similar to modern Egyptian populations, with some interesting variations that hinted at population movements and mixing over the intervening millennia. The ancient world was not static. People moved, populations mingled, genes traveled along trade routes and military roads and river valleys. Blood type was always part of that traveling, carried invisibly in the bodies of migrants and merchants and soldiers and pilgrims. The Silk Road, that ancient network of trade routes connecting China to the Mediterranean, was not only a path for silk and spices, it was a path for genes. Populations along the Silk Road show the genetic signatures of centuries of mixing, and blood type distributions along that corridor reflect the complex layering of populations from east and west that trade and movement produced. The relatively high frequency of blood type B in Central Asian populations may be, in part, a legacy of those ancient exchanges, the biological residue of a world that was always more connected than it sometimes seemed. Do check out the rest of the playlist if these ancient journeys are calling to you. There are many more stories there, each one a different doorway into the quiet science of the world. And if this particular night has been a good companion, a like on the video carries that kindness forward to other tired listeners who might need it. Let us rest for a moment in the present, not in ancient history, not in the future of research, but right here, in the particular softness of tonight. You are lying somewhere comfortable. The day is behind you, the night is around you, and inside you, unnoticed and unfailing, the quiet work of your body continues. Your red blood cells live for about 120 days. During that time, each one travels through your heart and lungs and every organ and tissue in your body hundreds of times, carried by the force of your heartbeat, navigating through vessels large and small, doing its work of carrying oxygen and releasing it where it is needed. At the end of their 120 days, old red cells are recognized as aged by the spleen, that soft, fist-sized organ tucked under your left ribs, and gently removed from circulation. The iron from their hemoglobin is recovered and recycled. The rest is broken down and excreted. It is an elegant, almost ceremonial process, the body honoring what has served it, returning what can be returned, releasing what is no longer needed. The spleen is also an important part of your immune system, a reservoir of white blood cells and antibodies, a filter that catches bacteria and other pathogens from the blood. People who have had their spleen removed for medical reasons, a procedure called splenectomy, sometimes necessary after injury or in certain diseases, can still live healthy lives, but they may be somewhat more vulnerable to certain bacterial infections because the spleen's filtering and immune functions must be compensated for by other parts of the immune system. The body adapts. It always finds a way to continue. Red blood cell recycling happens continuously, invisibly, 24 hours a day. Right now, even as you rest, your spleen is quietly doing this work. reading the surface of red blood cells, recognizing the ones that have grown old, drawing them gently out of circulation. The iron they carry is passed back to the bone marrow, which uses it to build new hemoglobin for new red cells. A cycle with no beginning and no end, running through your whole life. There is a condition called hemolytic anemia, in which red blood cells are destroyed more quickly than the bone marrow can replace them. This can happen for many reasons. Autoimmune attack, genetic abnormalities in the red cell membrane, certain infections, or transfusion reactions if blood type is mismatched. The symptoms, fatigue, pallor, shortness of breath, reflect the body working harder to compensate for the reduced oxygen-carrying capacity of its blood. Doctors treat hemolytic anemia in different ways depending on the cause, sometimes with transfusions, sometimes with medications, sometimes by addressing the underlying condition. In every case, the goal is to restore the balance, to bring the production and destruction of red cells back into their quiet equilibrium. Sickle cell disease, which we touched on earlier, involves a form of hemolytic anemia because sickle-shaped red cells are more fragile than normal ones and are destroyed more quickly by the spleen. People with sickle cell disease typically have a red cell lifespan of only 10 to 20 days compared to the normal 120. Their bone marrow works at an elevated pace to compensate, producing new red cells as fast as it can. This constant high demand on the bone marrow is part of why sickle cell disease is so exhausting to live with. The body is perpetually catching up. And yet, people with sickle cell disease do live. And many live with remarkable courage and adaptation, managing their condition with medications, transfusions, careful attention to triggers, and increasingly with newer therapies, including gene therapy, which holds real promise for treating the underlying mutation. The story of sickle cell disease is, like the story of blood types generally, a story about the body's complexity, its vulnerabilities, its resilience, and the gradual, patient work of medicine to understand and support it. Let your breathing be easy now. Let the stories of the body settle around you like something warm and familiar. None of this demands anything of you tonight. You are simply listening, drifting, letting the science wash through you in the gentlest possible way. There is a quality of wonder that comes not from dramatic discoveries, but from simply paying close attention to ordinary things. And blood is one of the most ordinary, extraordinary things in the world. Every person you have ever met, every person you have ever loved, has been sustained minute to minute by this same quiet system. Blood typing and flowing, cells aging and renewing, hearts beating their 100,000 beats a day. The person sitting across from you at breakfast, the child you tucked in tonight, the stranger on the other side of the world who is also, right now, lying in the dark trying to sleep. All of them held up by blood, all of them carrying their ancient blood type on every red cell, all of them connected by this shared biological inheritance to the same deep human story. We are far more alike than we are different. Blood type variation is real, and it is fascinating, and it carries within it the whole long history of our species. But it is a variation on a single theme. The theme is the same for all of us. Blood that carries oxygen, a heart that pumps it, cells that need it, a body working with exquisite complexity to sustain a life. Your blood type is a detail, a beautiful and storied detail in a picture that is overwhelmingly shared. Sleep has that quality too, of being deeply shared. Right now, as you drift toward rest, millions of other people around the world are doing exactly the same thing. Different time zones, different rooms, different blood types, different languages dreaming in their heads, but all of them surrendering to the same ancient need, all of them letting the day go, all of them trusting the dark to hold them until morning. You are never alone in this. The night is full of sleepers, and you are one of them, and that is a quiet and lovely thing. If you would like to share this feeling with someone you love who struggles to sleep, the playlist is full of nights just like this one. And a comment below, even a single word, is always a welcome thing to find in the morning. One of the softer and less discussed aspects of blood type science is its role in forensic anthropology, the study of human remains to answer questions about identity, ancestry, and history. Forensic anthropologists working on mass graves from historical conflicts, on the remains of individuals found in unusual circumstances, on the bones of unidentified people found decades after their deaths. These researchers use every available tool to learn what they can. Blood type, when it can be determined from bone or tissue samples, is one small piece of the puzzle, not definitive on its own, but useful in combination with other evidence. Age, sex, stature, isotope ratios that indicate what region a person ate and drank in, mitochondrial DNA that traces maternal ancestry. There is something quietly respectful about this work. The careful science applied to people who can no longer speak for themselves, in the hope of returning to them a name, a story, a place in history. Blood type as a form of witness. The antigens on cells that are centuries old, still faintly readable, still carrying information from a life that was fully lived. The oldest confirmed blood type analysis from an ancient human sample involved tissues from Egyptian mummies, and those findings helped researchers map the genetic continuity and change of Nile Valley populations across thousands of years. More recently, advances in ancient DNA technology have largely superseded blood type analysis for historical samples, since DNA provides far more information. But blood type was an important early tool, and the history of what it helped reveal is a quiet chapter in the larger story of how we have learned to read the past. There is also a living tradition of using blood type in genealogy, the study of family history. Before DNA testing became affordable and widespread, blood type was sometimes used to trace family relationships or to rule out certain biological connections. If a child's blood type was impossible, given the blood types of the claimed parents, that was a significant piece of information. Today, DNA testing has largely replaced blood type in genealogical contexts, offering far more resolution and detail. But for people whose family histories were shaped by the mid-20th century, when blood typing was the most accessible genetic test available, blood type records, sometimes preserved in hospital documents or military files, are still occasionally useful clues in the puzzle of who came from whom. Genealogy is, in a way, a personal form of the same ancient story we have been following all night. The story of how genes move through time, how traits are inherited and modified and passed forward, how the present is always the product of an inconceivably long past. Your blood type is your own small chapter in that story, and the person you might someday pass it to, a child, a grandchild, someone whose name does not yet exist, will carry it forward into a future you can only imagine. That is enough of a legacy for any molecule, to be passed forward, to keep going. The night is very deep now, and you are very close to sleep. Let the ideas become softer, less defined. Let the words dissolve into rhythm, into the quiet pulse of something that asks nothing of you. There is one last thing worth holding lightly before you go. A thought about what blood type research represents in the larger story of science. It is a story about careful observation, about noticing something small and strange and following it patiently. Karl Landsteiner noticed that blood samples clumped when mixed in certain combinations. He could have dismissed it as a contamination artifact. Instead, he followed the observation systematically and methodically until it yielded one of the most important discoveries in the history of medicine. The entire edifice of modern transfusion medicine, the millions of lives saved by matched blood transfusions, the blood banks running quietly in every major hospital in the world, all of it traces back to one careful scientist noticing something odd in a test tube and deciding to understand it. Science, at its best, is like this, a quality of attention that refuses to look away from the unexplained, that trusts the small and strange and unglamorous detail to lead somewhere meaningful. The history of blood types is a perfect example. What began as a curiosity about why blood sometimes clumped became, over a century, a window into human evolution, prehistoric migration, immunology, pregnancy medicine, forensic science, and the deep history of our species. One thread, pulled gently and with patience, unraveling into a tapestry. And the tapestry is not finished. It is never finished. Every generation of scientists finds new threads, new questions at the edges of what is known, new methods that make the previously invisible visible. The story of blood types will continue to be told, enriched, refined, for as long as humans are curious about themselves, which, if the past is any guide, will be a very long time. You carry a piece of that story in your body right now, not in your mind, not in your memory, but in the cells themselves, in the specific pattern of antigens on the surface of your red blood cells, in the particular version of the ABO gene sitting on your chromosome 9, in the ancient, inherited, faithful language of your biology. You do not need to think about it. You do not need to do anything with it. It is simply there, as it always has been, as it always will be, flowing through the night with you. Let your mind go quiet now. Let the science dissolve into warmth. Let the warmth dissolve into something that has no name, just the feeling of being held, of being safe, of being exactly where you are supposed to be. Your heart is beating, your blood is flowing, your body is resting, the night is holding you. Sleep now, sleep easily and deeply, and know that whenever you need another quiet night like this one, this channel and this playlist will be here, patient as a river, always flowing, always ready to carry you gently toward morning. Good night, and thank you for being here